All right, everybody, let's get this program started. Good afternoon, folks. Welcome to the Lunchtime Discovery Series. It's good to be with you again for another edition of our program where we get to meet some interesting people, hear about the work they're doing to help us learn more and share more about the natural world, science, the environment, conservation, education. We tackle a lot of different topics here on the Lunchtime Discovery Series. Every Wednesday at noon, I hope that you join us. I hope you've got your calendars marked. I hope that you've signed up to get that email newsletter about this lecture series so that you've got the links in your inbox every Wednesday ready to join us because we talk about some really cool stuff here, and there's a lot of cool stuff to continue to talk about. And if you're getting that email newsletter, you'll get updates every month on what presentations are coming up in advance, so you can mark the ones that you know for sure you don't want to miss. And you'll have the link handy so you can bookmark our YouTube channel and come back here every single Wednesday. Now, for today's program, we are not so far from home and at the same time quite far from home in lots of different ways. Today's two guest speakers are bringing the Carolinas together for some exciting scientific research. Let me introduce our guests. The first person that I want you to meet actually works for the North Carolina Museum of Natural Sciences as the research curator for non-molluscan invertebrates. That's Dr. Bronwyn Williams. Hi, Bronwyn.
Hey, Chris.
Thanks for being here.
Yes, thank you for having me.
Or I guess I should say thanks for being there, which looks like a gorgeous setting.
It is. I am currently in Eastern Texas in Big Thicket National Preserve. Wholly unrelated to crayfishes, but in a weird way, actually very central to the story that we're going to be talking about.
Save that one for the slideshow then, I guess.
I think so.
And then everybody, I'd like for you to meet Dr. Michael Kendrick. Michael is the assistant marine scientist for crustacean research and monitoring with the South Carolina Department of Natural Resources in the Marine Resources Research Institute. Hi, Michael.
Hey, Chris, thanks for having me.
Yeah, glad you could be here. This is exciting that we've got North Carolina, South Carolina, and weirdly Texas all represented in today's Lunchtime Discovery program. Which, folks, let me remind you, this program is a service of the North Carolina Office of Environmental Education and Public Affairs within the Department of Environmental Quality, and then it's broadcast and hosted by us here at the Museum of Natural Sciences. So I think I checked all my boxes. Let's get started and learn about threats to native crayfish and the battle for species survival. Well, we get set up. Bronwyn, is it warmer in Texas than it is in North Carolina today?
I don't know. It's pretty chilly here. We started in the 40s. So what's it like there?
It's about the same.
Okay. Yeah, I'm not sure that I planned on this when I thought about doing this talk outside and on the preserve, but it's all good. So, all right. Thanks, Mike. Okay. Well, I'm very much excited to team up with Mike Kendrick to do this talk, kind of in part on a project that we've been working on, or I've been helping with in the last several years. About a topic that's a little bit near and dear to my heart. So for those of you that don't know a heck of a lot about crayfishes worldwide have about 650 approximately, give or take, species, nearly 2/3 of which are found in North and Central America. And the highest diversity is in the Southern Appalachian region, which is great for us because North and South Carolina fall right kind of at the edge of that area. Okay, next. But of those crayfishes, we're actually focusing on a particular genus, a particular group of crayfishes, genus Procambarus, which happens to be the most species-rich genus of crayfishes in the world. It has almost 170, if not more than 170 species, all native to North and Central America. So this is the distribution that I pulled from GBIF. So essentially, this genus ranges from the south, from Guatemala and Honduras up along the Gulf Coast, although there is a little bit of pocket in Mexico on the Pacific slope, which is quite unusual. You've got along the Gulf Coast, and then you have fingers, one reaching up the Mississippi River basin, and then another one that reaches up the East Coast. So this genus of crayfishes, I mean, it's highly diverse, obviously, 170 plus species, but it's highly diverse, not just taxonomically, but also ecologically, behaviorally, such. So you've got stream dwellers, swamp dwellers, pond, vernal pools. You have primary burrowing Procambarus species. You've also got troglobitic, so cave specialist species. So high diversity in what they're doing on the landscape, where they're found, what they look like. And not surprisingly, this has led to a lot of confusion in what constitutes a species. And then in turn, kind of how these various species are related to each other. Okay, next. So we want to focus in on our topic today, which is North and South Carolina. And we've got a total of 19 total Procambarus species that are found in these two states. 8 species, which are found in North Carolina, 17 that are found in South Carolina. For those of you that want your checklist, these are all 19 species. The N is going to be those that are found in North Carolina, S found in South Carolina. North Carolina, we've got 2 species all to ourselves. South Carolina has sort of more than that. We share a whole bunch of them. But of this count, there is one, it's at the very bottom of the list, Procambarus clarkii, which Mike Kendrick is gonna get back to kind of at the end of this story. The rest of them are native. So we've got 7 native Procambarus species in North Carolina and 16 native species in South Carolina, and then this one non-native. Okay, so it looks like, again, this is all information. These points represent data that I pulled from GBIF, sort of this international collective of collection information and such. The blue dots are those from GBIF. The orange is what I've supplemented from samples that we've done over the last several years that haven't been published yet in our database. So if you look at this and you think, wow, that's a lot of sampling of, of this genus Procambarus, you must know a lot about sort of these species. And indeed, we know that there are 19 species of Procambarus on the landscape. But that's a little bit more of kind of this questionable, questionable situation. So let me dive into this a little bit. So Procambarus, just in general, as I stated before, is taxonomically difficult. So we've got these high diversity of ecologies and of where they're found, how they're found, what they're doing. And as a result, you've got a high diversity in the morphologies. But here we don't have many of those. We don't have cave dwellers in the Carolinas. We don't, sort of in the Procams, we don't have much in the way of the primary burrowing Procambarus crayfishes. What we do have a lot of are these stream dwelling, bog dwelling, swamp dwelling species, which have all converged on kind of very similar morphology. So this has led to a lot of taxonomic confusion. So you're seeing a lot of these different species that to me always look like little torpedoes with big eyes and narrow claws. So they're adapted basically to be living in the sluggish water area. And take a kind of that 100-mile view, step back. I mean, Hobbes in 1984 had already acknowledged that the genus Procambarus was tricky. And so, was saying that, at the time of writing, give or take, that sort of really at that point, only then could a range of a single species of Procambarus be assigned to that genus, could be more than vaguely circumscribed. And even now, we do not know the limits of ranges of most species. And I think the same can be said, even despite all this sampling, we know that there are Procambarus crayfishes everywhere across the Carolinas. But if we look at these dots and we start to split them into species, we're not exactly sure where one species ends and another begins. So this was kind of the whole premise for some of the work that we were doing. Okay, next. So here's just an example. I mean, of several of the species or 6, I guess, of the species that we find in North and South, Procambarus species that we find in North and South Carolina. And when you look at the slide, that gut feeling is, oh, well, these are clearly 6 different species. But if I remove the color from many of those species, let's say we collect them, they go in museums, that color is removed, they're just kind of bleached out, specimens, that kind of those differences start to go away. There's also a lot of variation within species. So when we take these photos, we typically try to, take kind of the most showy and exemplar specimens that we can. And the other thing to think about is that when we're in the fields collecting these specimens, a lot of times we're picking up lots of little tiny little juvenile crayfishes, and a lot of those are what end up kind of creating those points that we see. And we're just kind of like, I don't know, this could be species X, species Y, I don't know. Basically, what I'm trying to get at is that sort of the taxonomy of this genus has been historically tricky. Essentially the gods of crayfish taxonomy have acknowledged that. And certainly I think Mike would probably agree with this, is that spending a lot of time in the field collecting them, it's no less easy. And I think kind of being a crayfish biologist, we learn, we kind of have to acknowledge that the more we learn, I feel like the less we know. And Procambarus species in the field are certainly a good example of that. Okay, next. So one of the things that we were really interested in doing was saying, okay, we know that sort of Procambarus crayfishes are a major component of the crayfish diversity of both North and South Carolina. So in North Carolina, we've got 9 Procambarus species out of about 50 total. And in South Carolina, we've got these 16 or 17 Procambarus species out of a total of about 36. So South Carolina Procambarus species make up the majority, or almost half, of that total sort of state species diversity. A little bit less so in North Carolina. But if we think about the coastal plain and the lower Piedmont, Procambarus are the dominant organisms in those areas. So what we've been doing, and we continue to do, is look at understanding or trying to get a handle on native biodiversity using an integrative approach. So we can use what's been established as the morphological based species identifications or diversity as a baseline. We go out, we sample systematically for whatever we can find throughout the landscape, the coastal plain, the Piedmont. We then use genetic methods to essentially confront the morphological data. And then we can use the results of those, the genetic analyses, to revisit how we're looking at the morphology. In addition, we can add other data sets like behavior, biogeography. So where on the landscape or in these various drainages are these organisms found? Color patterns certainly important where we can find it. So what this sort of very potentially complex and pretty mobile looking figure is showing you the preliminary results of some of the genetic work that we've been doing with Procambarus in North and South Carolina. So I've stripped all of the names, thankfully, off of this circle tree, but this is about 400 individuals that were used to create this phylogeny. And really, one of the major take-homes that we discovered from this was that genetics is very useful for distinguishing species. So this may sound kind of like a duh moment, but with coastal plain species, we were a little bit concerned, given that you have cyclic flooding along the coast, that perhaps so these various species that get down into the coastal plain areas would be cyclically mixing back and forth. So you would actually see this genetic swamping and its dilution effect within what were called distinct species as you moved along these gradients from kind of higher up in the drainages down in the coast. And maybe this was just my expectation, but it was a concern that we might not actually have these clear-cut species. Well, what we find from the work that we've done so far is that's not the case at all. And in fact, genetic data are, I mean, beautifully supporting kind of what our concepts of species are, and are helping us, you know, sort of providing us with additional insight into diversity that we didn't even know was there. So long story short, genetics have proven for this group to be, for us, have shown to be very, very useful for the work that we're trying to do. Okay, so what are some examples of what I'm getting at here? Because that seemed very abstract. All right, so Procambarus acutus, and I'm sort of sitting in the heart of in Texas here in acutus territory. It's this hugely wide-ranging species that is found basically all along the Gulf, kind of up through the Mississippi River Basin, and then up the East Coast, all the way up into Long Island Sound, basically, sort of New York, New Jersey area. So we already knew, kind of going into this, that you've got this huge, broad, wide-ranging species, and it happens to be one of the most wide-ranging species in North Carolina, not so much in South Carolina, but certainly in North Carolina. It's a little bit of a dumpster fire, if I kind of say that. It's our trash taxon. It has so much variation that doesn't appear to anybody to have any geographic cohesiveness. And so as a trash taxon, there are a lot of folks, myself included, that when you're out in the field and you kind of go, well, I don't really know what that is. That looks kind of like acutus. It gets acutus as a name on it, and away it goes. Well, what we discovered, which perhaps may not be shocking given kind of how broadly distributed and everything that this is, that acutus is actually showing to be two very, very clearly different genetic groups. And in fact, we've got within those genetic groups, we've got substructure that may or may not be at the species level. So those may be at the level of, we have these very distinct populations or evolutionary significant units within each of these. But at this point, it looks like Procambarus acutus in North and South Carolina is at least two different species that we have yet to kind of determine. So we can use these genetic data to then again go back and look at the morphological information that we've already gleaned, to kind of confront that morphological data in a geographic context to try to figure out, does this make sense? How does this make sense? Can we describe these species so they can be adequately conserved? Okay, so that's one example of what we've been doing, taking a kind of a broad-ranging species that doesn't have any sort of conservation concern in either of the states right now, and saying, we've got to look a little bit more closely at this. Next. Okay, so now I'm going to kind of go to the flip side of that and say, all right, well, what I didn't sort of mention when I was talking about the different Procambarus species that are found in South Carolina is that almost all, with the exception of Procambarus acutus in North Carolina, almost all of the Procambarus species are of conservation concern in North Carolina to some extent. So we have some endangered species, we have threatened species, we have several that are on the watch list. Very, very few are being considered stable. In South Carolina, we have several species that are effectively kind of on the conservation list, sort of this watch list that they have to some extent. Okay, Procambarus ancylus is one of these species that is of conservation concern. So we know kind of very little about the distribution of this species, despite all that sampling in this area. There are sort of impacts of things that are a little bit outside of our control at this point that we're trying to get a handle on, that are going to be impacting the stability, long-term stability of this species. So we wanted to look at this critter a little bit more closely. And what we found, again, using our integrative approach, was that Procambarus ancylus So what you see on the map on the right-hand side, these dots are the ones that we used to create the tree on the left. So essentially it's a one-to-one. So it's not all ancylus that we know about, it's just the ones that we were able to sequence and then use as other data for. Long story short, Procambarus ancylus, which is already of conservation concern, is actually two different species. So the type locality is what is sort of near where that blue star is, down near Charleston, sort of actually Somerville area. So this is something that actually that Mike and I are going to be working on, kind of moving forward over the next year or two, to really suss out what are the distributions of these two different taxa within this group. Okay, next. And the last one that I want to leave with you before I turn it over to Mike is a taxon that has very close ties to the North Carolina Museum of Natural Sciences, Procambarus braswelli, who was named after Alvin Braswell, sort of former herpetology curator, now emeritus. A lot of folks watching this have probably interacted with Alvin in some way, shape, or form. Well, Procambarus braswelli, which was described from Waccamaw River, just south of Waccamaw Lake in North Carolina, is considered endangered in the state of North Carolina, and it's considered of great great conservation concern in South Carolina. Well, we were a little bit shocked to get the genetic data back that showed that Procambarus braswelli is basically indistinguishable, genetically indistinguishable from Procambarus chasei, which is found kind of around the Charleston area. The type locality of that, the blue triangle, or so the blue star to the left, is going to be just near or just outside of Columbia, South Carolina. So, well, what does this mean? Well, it's kind of a good thing. I guess, when we saw this result, we were able to go back and look at, morphologically look at Procambarus chasei and Procambarus braswelli, and indeed they are indistinguishable morphologically as well. So, as I said, the good news is that we've actually just greatly sort of expanded, or as we work this through over this winter, hopefully, we've taken two kind of very endangered species, and we've just expanded the range of this now one synonymized species, which is a good thing from a conservation perspective. Although I think both of us would urge a very conservative approach on sort of stripping away any protections of what will be this one species because of a number of threats that it's facing currently and in the future. And with that, Mike, I'll turn it over to you.
Thanks, Bronwyn. That's great. I think that's a really wonderful overview of the native crayfish biodiversity that the Carolinas sort of collectively share. So I appreciate you going over that and providing that for the folks listening in. I do want to talk about at least one of the threats that is posed for this native biodiversity that we have in the Carolinas, and that's invasive species. So many of you may be familiar with invasive species, but it doesn't hurt to sort of go back over some of the generalities that we need to sort of understand when thinking about invasive species. And that is that they spread globally. You can see in the sort of image in the background here, these connection points between ports across the globe that can be really important for transporting invasive species globally. But we also recognize that invasive species can be spread on much smaller local scales as well. Invasive, we can start by sort of defining what invasive species are, and those are basically species that are not native to a particular environment that cause harm to natural resources. And when they cause harm to natural resources, they ultimately have some economic cost as well. Some estimates suggest that it's upwards of almost $27 billion per year that invasive species end up costing, and that includes their impacts on sort of losses to goods, services, and production, but also the cost of actually trying to go in and manage these invasive species and try and sort of prioritize how to mitigate the impacts of invasive species on natural resources. And part of that understanding is looking at the ecology of these invasive species. How do they interact with their environment? How do they move through the environment? And what are their impacts, and maybe in this case, on native biodiversity? And with those things together, we can help inform effective management strategies to mitigate their impacts. So we've learned about our native crayfishes. One of the invasive crayfishes that is in the Carolinas is the red swamp crayfish, Procambarus clarkii. And this is really a global invader. So this is a map here from a publication from a couple of years ago showing the global distribution of this species. We can see in North Carolina, there's a little bit of blue area on this map, and that indicates the native range of this species. They're native to the Gulf coastal plain, parts of Texas. The red on this map indicates the areas where this species has invaded and become established throughout South America, Africa, Europe, and Asia. A little bit on the movement of the species. Why is it spread across the world? Some of you may have participated in crawfish boils in the past and recognize that this is a species that can be an important source of food. And so there's aquaculture industries around the world that farm this species in part for that purpose. The species can also be used for bait and also in the aquarium trade. Once found in a particular environment, natural dispersal patterns and movement patterns, both in water, but also the species is known to come out of the water and crawl over land, over roadways, through forest to find new areas and new habitats. The impacts of this species, including the fact that they can displace native species. They can outcompete and displace native taxa and reduce native biodiversity. They're omnivores, so they can feed on macrophytes or large aquatic plants, and reduce the density of those aquatic plants. And they can also serve as vectors for parasites and diseases. And this has been particularly important for areas of Europe that have experienced what's known as the crayfish plague, that has caused the decline in a number of native crayfish species in Europe. So I have a couple of objectives from the project that I'll be talking about today, and that's one, documenting the spread of this species in the Carolinas, and then using some molecular tools to try and infer dispersal patterns, and then do a little bit of sort of overview on how this species may actually be impacting some of our native crayfishes. So the first part of this exercise is sort of compiling historical crayfish data. What do we know about crayfishes in the Carolinas? Dr. Williams did a great job showing a broad scale across the Carolinas. What are those distributions of crayfish data? The project that I'm focused on here is focused on the PD River Basin, which is what I have highlighted here. You see this sort of line running diagonally across the map. Here is the border of North Carolina and South Carolina. In purple is the Waccamaw drainage. Green, we have the Little PD drainage. And then in the sort of beige color, there is our lower PD. And what I'm showing here, records of native crayfishes in white that were collected prior to 2000, sometime within the 1900s, including some of those that go back to as early as 1905. And then we also have records of our red swamp crayfish, Procambarus clarkii, that are shown in red here. And we can see those are isolated to the bottom of the page, suggesting that right during the 1900s, the distribution of Procambarus clarkii was restricted mainly to this area of the extreme lower Waccamaw and the lower PD basin. And then in part, some of the fun that we get to do for this, for these sorts of projects is collecting new data, getting out into the field, getting out to some of these locations throughout that the PD drainage, and collecting information on the current distribution of crayfishes. We use a number of different techniques. We put out basically these minnow traps to trap crayfishes. We often use dip nets. We'll dig burrows. So that's me on the top right with my arm in what was a hole in the ground. I reach in and pull a crayfish out. And also backpack electrofishing, which can be an effective way to collect crayfish. So we are in the field across this watershed collecting data on crayfishes, where they're found. We get to experience lots of fantastic habitats within the Carolinas. They have a lot to offer, especially within the coastal plain areas for which most of this work was focused. Creeks, streams, and rivers. These are the places that we often think about as being sort of quintessential crayfish habitat. Some sandy bottom, some filled with cypress trees. These are some of the habitats we go to. There's also roadside ditches, which is a habitat that I feel is underappreciated, but maybe becoming more appreciated as of late. These are in some ways sort of vernal pool type habitats and ephemeral habitats where they fill with water sometimes, especially right after rains, and they can dry out at other times when the crayfishes can actually burrow down into the sediment and withstand that period when there's not actually any surface water there. Similarly, we go into swamps that can have a similar sort of hydro period. These are swamps that have very large and old cypress trees and cypress knees, as you can see on the left here, and other swamps that are inundated at times of year, and sometimes you go out and it appears dry. But oftentimes, again, these habitats, there are crayfish that are living right under your feet, and we just don't quite realize it. So as part of this project, we go out to all of these different habitats as we can and collect information on the numbers and types of crayfishes that we find. After that, we come back into the lab and we can process those data and analyze those samples. And then we can document the spread of this species. The map I'm showing on the left is the map I showed a few moments ago. In the 1900s, our distribution of Procambarus clarkii in red was restricted to that sort of extreme lower Waccamaw and lower PD region. We contrast that with the new map here on the right, which is all of the collections from the 2000s, and including the collections for this project. And we can see that the Procambarus clarkii is now very widespread across each of these watersheds. And so this is really indicating or suggesting to us that this species has spread fairly rapidly throughout the 2000s. And we have other data to suggest that this has been a fairly rapid spread. One important point here is it's not that we go out to these sites and we find one of these specimens. Oftentimes we have very high numbers of these non-native species of the red swamp crayfish that are found at these locations. And the picture of the bucket on the right is an example of that. That's a bucket that is filled with Procambarus clarkii from a relatively short sampling event. So this species is not only very prevalent, it's present at many different sites across these watersheds, but when it's present, it is often very abundant and usually in greater abundance than the native species that are there. So we have a lot of information here, but one of the things that we want to learn more about is this sort of rapid spread of this species. How did it happen? Why did it happen? How is the species sort of moving so quickly across the landscape? And so we can ask a separate set of questions and sort of inferring and understanding sort of movement or dispersal patterns of this species. And we've developed molecular tools, including some genomic sequencing, in effect allow us to ask the question of, is the genetic structure of just this species of Procambarus clarkii, is it related to these watershed characteristics? So again, this map of our three sort of key watersheds that we're working with, and we sort of developed these sort of longitudinal sites, 1 through 7 or 1 through 8 across the Waccamaw subbasin, as you can see, starting up at sort of Lake Waccamaw is number 1 there, and sort of moving downstream from that. Similarly, in the Little PD Basin, starting 1 at the sort of upstream locations and moving downstream. Each one of those sort of white circles there is location where we collected the red swamp crayfish from that site, and then went back to the lab with our population genetics section here at the South Carolina Department of Natural Resources to look at these molecular tools to help us ask this question of how the genetic structure and the relatedness of these different sort of subpopulations of Procambarus clarkii and whether or not that's related to watershed characteristics. So I'll try and go a relatively sort of quick overview of these results. These are called structure plots, and I could talk for a long time about how to interpret them, but to try and give you sort of the high level view there, I have separated on the bottom here sort of 1 through 8 sort of groups, and each of those represents the 1 through 8 that are listed in white circles on the map, just from the Little PD subbasin there. And what you see from that is we basically have some that are blue and some that are green. And what that represents is that we sort of had these two genetic clusters, or these two genetic groups that suggest that those ones that are all blue are more related to each other, and the ones that are all green are more related to each other. So it's indicating that there's not a lot of sort of structure, and that there's probably a fair amount of gene flow between these different sites. All the ones from 3 through 8, from 3 downstream to 8, there seems to be a lot of gene flow across those. So there's some connectivity suggesting that there may be somewhat rapid spread of those organisms across that relatively wide spatial scale. Similarly, when we look at this in the Waccamaw Basin, we can see a few more colors here, but we still see a lot of similarities, a lot of the same colors, greens and blues that are in there. Again, suggesting that there's a lot of gene flow between a number of these sites. And some of that is moving sort of in that downstream direction. So in some ways, the way that we're interpreting these results is that there seems to be potentially a fair amount of natural dispersal in that sort of upstream, downstream direction for both the Little PD Basin as well as the Waccamaw Basin. Now we can contrast that with the Lower PD Basin. What we see here is that each one of the sites here basically shows up as a different color in the structure analysis. And that suggests that there's not a lot of gene flow between these different sites. Now, if you notice the Little PD Basin, the sites in this Lower PD Basin, they're fairly close together. So we would have expected a fair amount of gene flow between these sites, but the results suggest that there's not a lot of gene flow. Some of this may be due to the fact that, as we saw in the earlier figure, this species has been established in this area for quite a long time. And some of that has been in part contributed to the aquaculture of this species that was conducted in this region for a number of years before Hurricane Hugo sort of took that industry out. And what we see here is that despite the close proximity, there's a lot of structure genetically. So again, this may be a sort of carryover from that historical aquaculture industry, or at least having these sort of long established populations here that prevent that gene flow between sites. So the dispersal mechanisms, what can we sort of take away from this? Well, the results do seem to suggest that there's natural dispersal that is occurring within the Waccamaw and the Little PD basins, especially. Some of this is likely related to these longitudinal patterns of river flow. There doesn't seem to be very many sort of introduction points, right? There's not a lot of indication that these crayfishes are being sort of intentionally introduced into lots of different areas of the watershed, but maybe a few introduction points that have then led to sort of rapid natural dispersal. We do see, particularly in the lower PD basin, that variability that's unrelated to the watershed structure, and seems to be potentially associated with the historical aquaculture activities in the region. So what does this mean for our native biodiversity? Coming back to sort of the starting point of this presentation, what are the potential impacts of the red swamp crayfish on native species in this region? So again, come back to this same map. Hopefully you're familiar with it now. We have a couple of different sort of color-coded points here. The white are the points where we have our Procambarus braswellii or Procambarus chasei, the Waccamaw crayfish and the Cedar Creek crayfish, locations where those species have historically occurred. In gray are the species where we have records of those of one of those species or that species as it may be, but that is also a location where it is co-occurring with Procambarus clarkii. So these two species are collected within the same river system, collected together within the same system. That raises lots of sort of concern for how well that native species may sort of withstand that invasion. In red, as we have the 3 points in red here, these are sites where we historically have had records of this species, but now the only species at that location is Procambarus clarkii, is the invasive red swamp crayfish. And we can sort of label that as a site where the native species have been potentially extirpated from that area. Our sort of collective concern is that we have these areas of co-occurrence, and this species may be able to sort of withstand and continue to co-occur with the red swamp crayfish. But if these red points are any indication, this native species could be extirpated from these additional sites as well. And so wanting to maintain our sort of native biodiversity, this is something that we're certainly looking at closely, that we can try and help mitigate. We have a very similar figure that we can look at for Procambarus pearsii. Again, we have at least 3 locations that have been, the species has been potentially extirpated due to the presence or likely due to the presence of the red swamp crayfish. A number of other locations where we have Procambarus pearsii and not necessarily records of the red swamp crayfish, and certainly some records where these species do co-occur. So this is showing us that there's some indication that Procambarus clarkii is potentially extirpating some of our native crayfish species. So what can you do to help reduce the spread of invasive species? Many of you have been to crawfish boils, have experienced crawfish boils, or at least are aware of crawfish boils, and that's one of the ways that crayfish can get moved across areas. One of the things that we can do is to try and be careful and help minimize the escape artists when we partake in these sorts of activities. For those of you that have, you recognize that you have a cooler full of the red swamp crayfish in your backyard. They can very easily crawl out and crawl into the ditch in your front yard and quickly become established. So it's just being aware and being cognizant of these species, of these activities, and trying to prevent those escape artists, but also to minimize the movement of animals, especially for things such as bait. For using crayfish can be a very effective fishing bait. So being real sort of careful about moving sort of live animals across watersheds and across areas. If it's not native to a region, being very careful not to release those live animals into the wild. It's important to note that the work here has a couple of different funding sources, right? So internally for the South Carolina Department of Natural Resources, as well as with Dr. Williams, the North Carolina Museum of Natural Sciences, but also multiple grants from U.S. Fish and Wildlife Service were really central to conducting this research and getting this information out to you all so we can help understand how we can protect and conserve the natural resources of both North Carolina and South Carolina. So thanks for listening in today. Happy to take any questions that may, I think, show up in the chat.
All right. Fantastic job, Dr. Williams and Dr. Kendrick. I would invite everyone in the chat to drop lots of clapping hands emojis for everyone to show their appreciation for a really good presentation. Like, you covered it from the start to finish. Here's all of the really cool crayfish species, why they're important, and the amazing diversity that we have in our backyards in North and South Carolina, and then why they're in trouble, and then what people need to do about it, or what people can think about as they begin their educational journey for crayfish. So excellent stuff. Yes, folks, if you've got questions for our guests, drop them into the chat. I'm going to be looking to you in just a moment for your questions, but I'm going to throw it back to a little earlier in the presentation. Bronwyn, you were talking about how well the genetic information helps you to separate out all of the different crayfish species, even if they live in like the same watersheds and drainages and habitats like that. But I'm aware that within the biological sciences, there's this debate about how much genetic information needs to be different, what nodes or loci in the genes need to be different in order to actually declare a species new to science. Can you talk a little bit about what your process is to actually identify new species?
Sure, absolutely. So as I talked about, I mean, we're using an integrative approach, which basically means that we're not basing our definition of species on any one data set. So the genetic data, there's not really any one set I guess divergence level that we use. Essentially what we're looking at is for different taxa to be reciprocally monophyletic, so which basically means that they are distinct, genetically distinct from each other, whether that's 2% or 10%. It doesn't matter, as long as then those distinct clades are then backed up with additional support that we can find for morphology, for behavior, for reproductive behaviors, for geography. So it's basically this all-in evidence approach that we typically use. So there are certainly some folks that have a threshold if they're using genetics as the kind of overwhelming weighting factor for what they determine a species. There are some folks within the crayfish world that have suggested that 5% genetic distance, and this is going to be for the barcoding region of CO1, that's if it's below that, it's not a different species. If it's above that, absolutely. I do not ascribe to that at all. It essentially suggests that all speciation is occurring at the same rate and the same time, and we know that to not be the case. You know, there are things that are diverging much quicker than other organisms. Likewise, we're all at sort of different scales within this tree. Those nodes are appearing at different times and places. So again, I'm more of the let's confront a question with as many different data sets as possible, and then when we're comfortable kind of deciding that we can distinguish this as a different species, sort of in the field as well as in the lab and such, then that's when we make that move. But this also brings up a good point as to why do we describe species? So why is it important that we are out there, you know, sort of saying that there is new diversity on the land, you know, sort of on the landscape, and that we need to give it a name? Or with, you know, the situation of Braswelli, which is going to become synonymized with Chasei, why do we then kind of make this action to then make that synonymization. And that's something that I need to kind of remember to talk to in many cases. And the answer is really, it's not taxonomy is not an ego thing. So we're not describing species so that our name is kind of stamped all over these various things. It really is for the organisms themselves. So crayfishes in general, and Mike can certainly add to this. I mean, they are central components to aquatic ecosystems. If you think of a game of Jenga as being any aquatic ecosystem, any ecosystem really, but let's think of sort of freshwater aquatic ecosystems. You have different species of crayfishes that play very different roles. And so, if you were to start removing some of these native taxa, like playing Jenga, remove the blocks, you don't know what combination of blocks you remove before you end up with a whole collapse of that ecosystem. So things that crayfishes eat may become overabundant, take over these ecosystems. Things that eat crayfish may, then they don't have food, or they shift to something else that might be very critical. And then when we have these replacement, when we replace it with this non-invasive crayfish, they are not necessarily going to have those same interactions with what is in that ecosystem as the natives are as well. From a more practical standpoint, do we know that a particular crayfish looks at another one and says, you're a different species? Well, absolutely not. However, we can, again, using these integrative approaches, these different data sets and data types, we can make sort of our best, sort of indication of you are something unique. You are a unique component of diversity. And from a management side of things, we can't actually enact any conservation actions until that has a name. So species description is necessary in order for agencies to then take a stand to say, oh boy, okay, there's something, these X number of species in this watershed need to be protected. If they don't have names, that protection isn't there. So, Mike, I don't know if you wanted to add anything to that or not. We can move on.
I don't have anything to add, just to reiterate that the management side of understanding species delimitations really allow for much more effective management strategies that help us to sort of protect and conserve our natural resources.
Excellent stuff. Thank you. All right, so the next few questions that came in for you, there are several here. People want to know more about the red swamp crayfish, the clarkii. First one is from Susie. Is the red swamp crayfish the only crayfish that travels on land?
No, absolutely not. There are, I would guess most crayfishes will go walkabout if given the chance. And all that takes is actually high humidity. A lot of crayfishes, different species, primary burrowing species, stream dwelling species. I think, I've heard a number of tales of people doing, sort of along the walking path after a good rain and seeing a crayfish walking along that path as well. But again, even a good heavy rain, is not necessarily needed for that to happen.
Yeah, I agree with that. And I'll add that I've received sort of a number of reports from people that have seen sort of mass movements of red swamp crayfish, where they just see large numbers of this particular species sort of moving across land. So I think there's certainly questions as to how different species may use this sort of overland movement as sort of dispersal mechanisms. I think it's pretty common for lots of species, but some of them may do it on a more sort of individual basis, and others may do it in more sort of direct cues to sort of rain events or things that sort of facilitate a more sort of mass movement, if I can use that term there.
Yeah, okay. Well, that sounds like a sight to see. Just
right.
Dozens of crayfish coming up out of the water across the road. Cindy wants to know if the invasive red swamp crayfish can hybridize with any of the native species.
Yeah, that's a great question, and that's actually a question that we've had, and we've used some of our molecular techniques to investigate whether or not that's occurring. We do have a native species, which is called the eastern red swamp crayfish, is the sister species to Procambarus clarkii, and that one is native to South Carolina. So we've had concerns that when these species are co-occurring, that there may be some hybridization. So we've looked at the genetics of those sort of individuals that are co-occurring and don't have any evidence that they are hybridizing. So in some ways that may be a good thing that these species are, at least so far, all indications are that there's no hybridization sort of occurring between these different species. That's a great question, though.
Excellent stuff. Steve wants to know if this invasive species has made it into Lake Waccamaw.
Yes.
Yes.
Go, Steve.
Sorry, Steve. Yeah.
So, yeah, sorry, Steve. Okay. Have you noticed habitat differences between sites where the native species are extirpated versus those where there is still coexistence? Right, Susan?
Oh, no, that's fantastic, right? And I think that's where better understanding some of the natural history and ecology of our native species alongside our sort of enhanced understanding of this invasive species will really help us to better understand how, when, and where the coexistence may occur, and under what circumstances we may be more likely to see the extirpation. So I think it's a great question. I don't have data on that yet. Bronwyn may.
No, not yet.
Right.
On the way. Yeah, I mean, a lot of these questions seem to be getting this idea, what makes this particular crayfish, a super mud bug compared to our native species and their ability to disperse and thrive, across these rivers and streams and drainages. Yeah,
I think that is a question that can be addressed for lots of invasive species, right? I think there's a lot of different sort of ideas for that. For this one in particular, though, I think it's important to recognize that while the red swamp crayfish has expanded across the globe and into a huge range of different environments, I think the rapid spread of the species in the coastal plain environments may be helped by the idea that this species is native to coastal plain environments. It's native to coastal plain, the Gulf coastal plain, right? Louisiana, Mississippi. This is its natural habitat. Texas, right? So it's native. It knows these swamps. It knows the coastal plain. It's really good at living in that environment. And so moving over into the Atlantic coastal plain of South Carolina and North Carolina is a very similar habitat to its native environment, and I'm of the opinion that has sort of facilitated this somewhat rapid sort of expansion of the species in this area.
Do you think then that means that there are specific limits on its spread on the East Coast, temperature limits or something like that they wouldn't experience on the Gulf Coast and in the Gulf coastal plain? No, probably not. Going everywhere.
Well, I mean, I know of situations where the red swamp crayfish has been introduced into northern Canada. It was basically eradicated before they gave it a chance to establish. But I think that this is one of the tricky things with a lot of invasive species is that we don't really know what those thresholds are. Oftentimes, I mean, things that might make sense. Well, okay, if this is distributed in its native range here, maybe temperature is going to limit its expansion, and that's not necessarily the case. And of course, we don't really want to provide the natural laboratory to see what happens.
Yeah, sure. Excellent stuff. And just to make sure that we've got everybody on the boat with us, there was a question that came in. Crayfish look like tiny lobsters, and they have little tails like shrimps. So if you can, just in the last like 30 seconds that we've got, break it down, what is the difference between a crayfish and other things that look like crayfish?
Mike, do you want to take a stab at this as a marine sort of marine oriented biologist here?
There's crustaceans are such a diverse group. I would encourage anyone that is just trying to understand the difference is you have to taste them. You have to get some in your hands. You have to go shrimping, see what a shrimp looks like, see what a shrimp tastes like. Go get some crawfish and do the same. It's really sort of a hands-on experience, right? There's lots of sort of morphological differences, and we could explain that. But I think the best way to really experience what a different difference is to bring them into your kitchen.
That's amazing. But I guess from a little bit more practical standpoint, well, that's very practical, actually. I would say, yes, clawed lobsters are sister to crayfishes. So there is that similarity. They are kin.
They are kin, but they don't taste like it. That's a great that's the perfect answer to that question. If you want to know the difference, go eat them. That's the first time I've heard that on the Lunchtime Discovery series. I'm glad we got that question. And Susie Adams wants to buy your hat off of you, Bronwyn.
Susie, I can get you one. We got to support the hickory crawdads. North Carolina strong.
Oh, that's what that. It's
yes.
The like AAA affiliate.
Yep.
Okay. Minor league team.
Oh yeah. Don't worry, Susie. I'll get you one.
There we go. Well, this has been fun, insightful, very interesting. Bronwyn and Michael, thank you very much for being here.
Thank you, Chris.
Yeah, thank you.
Folks, we'll be back here again with another edition of the Lunchtime Discovery series. We won't be back here next week. We're taking next Wednesday off, but we will be back the first Wednesday in November on the 2nd with a talk from Michelle Jewell from North Carolina State University. Don't miss it. It's going to be great. Michelle's awesome. Make sure that you're following the North Carolina Office of Environmental Education on Facebook and on Twitter. You can get links to their social at their website, which is a great place to start for their resources. They're ee.northcarolina.org. And of course, you can get contacts for the Museum of Natural Sciences, our social, and see our calendar of events, which is full of great stuff for you to participate in at naturalsciences.org. Until next time, everybody, take care, stay safe. Don't throw any crayfish into the river that you don't know where it came from in the first place. And we'll see you again soon. Bye, everybody.